So last time I got started talking about macromolecules. I talked mostly about carbohydrate and lipids. Now let's resume what we start with. I'm going to cover the two other classes of macromolecules, proteins and nucleic acids. The importance of protein is underscored by their name, which comes from the Greek word proteos, meaning primary or first. They are instrumental in almost everything organisms do. Some proteins speed up chemical reaction while others play a role in defense, storage, transport, cellular communication, movement, or structural support. Life would not be possible without enzymes, most of which are protein. Enzymatic protein regulates metabolism by acting as catalysts, chemical agents, and you know that enzymes are selectively speed up chemical reaction without being consumed in the reaction. You have these 8 flashcards talking about the function of proteins. Please read them and you will see the importance of protein. You have 8 different functions for protein. Proteins are all constructed from the same set of 20 amino acids linked in unbranched polymers. The bond between amino acids is called peptide bonds. So a polymer of amino acid is called a polypeptide. A protein is biologically functional molecules made up of one or more polypeptides, each fold and coiled into specific three-dimensional structure. All amino acids share a common structure. An amino acid is an organic molecule with both amino group and carboxyl group. The small figure shows the general formula for an amino acid. At the center, you see the asymmetric carbon called alpha carbon. And remember that carbon can form 4 covalent bonds with carbon, with hydrogen, oxygen, or any other atoms. Its 4 different partners are an amino group. You have an amino group and carboxyl group, hydrogen atoms, and variable group are called the side chain. This group symbolized by R. The R group, also called the side chain, differs with each amino acid. Maybe simple hydrogen atom or maybe carbon skeleton with various chemical groups attached. The chemical and physical properties of the side chain determine the unique characteristics of particular amino acid, thus affecting its functional role in a polypeptide. This figure shows the 20 amino acids that cells use to build their thousands of proteins. Building a huge variety of polymers from such limited number of amino acids is analogous to constructing hundreds of thousands of words from only 26 letters of the alphabet. Let's see how they are linked to form polymers. When 2 amino acids are positioned so that the carboxyl group of one is adjacent to the amino group of the other, they can become joined by dehydration reaction with the removal of water molecules. The resulting covalent bond is called peptide bond. We call this type of bonds by removal of water molecule peptide bond. Remember that for the carbohydrate we call it glycosidic linkage, and in lipids we call it ester bond, and here between amino acids we call peptide bond, and the polymer we call it polypeptide. This bond is repeated over and over. This process yields polypeptide chains, a polymer of many amino acids linked by peptide bonds. So polypeptide is a polymer of many amino acids linked by peptide bonds. The repeating sequence of atoms highlighted in purple is called a polypeptide backbone. call this one the polypeptide backbone. And extending from it, those are the R or the side chain, the yellow and green boxes here. Those are the side chain extending from the backbone. Polypeptide range in length from few amino acids to thousands or more. Each polypeptide has unique linear sequence of amino acids. Note that one ends with one end had free amino ends, we call it N-terminus, and on the other side of the polymer, we have this amino acids ends with the carboxyl group, and we call this ends C-terminus. So we have two ends for each polymer. One ends, we call it N-terminus, that ends with the amino group, and the other end of the polymer ends with the carboxyl group, we call it C-terminus. When a cell synthesizes polypeptide, the chain may fold spontaneously, assuming the functional structure for that protein. This folding is driven and reinforced by the formation of various bonds between parts of the chain, which in turn depend on the sequence of amino acids. A protein's specific structure determines how it works. The function of protein depends on its ability to recognize and bind to some other molecule. Complementarity of shape between these two protein surfaces shows in this image the exact match of shape between an antibody, orange and blue part. Those are the antibody that's produced by the body as immune defense. And the particular foreign substance, this protein from the flu virus, the yellow and green parts. The antibody has complementarity shape with the protein parts of the virus flu and marks this virus for destruction. You may recall another example of molecules with matching shapes between endorphin molecule produced by the body and morphine molecules that manufactured by drugs. The endorphin that's produced by the pituitary gland, and morphine, this is a manufactured drug, both of which fit into receptor protein on the surface of brain cells in human, producing euphoria and relieving pain. In spite of their great diversity, proteins share three supermost levels of structure, known as primary, secondary, tertiary structure. The fourth structure level, quaternary structure, arises when protein consists of two or more polypeptide chains. The primary structure of protein is its unique sequence of amino acids. The primary structure, in turn, dictates secondary structure. We have alpha helix and beta helix, beta pleated sheet, and tertiary structure due to the chemical nature of the backbone and the side chain R group of the amino acids along the polypeptides. The amino acid indicated here by its three letter abbreviation. You see here the glycosine, proline, threonine, glycine, threonine, lysine, leucine, and valine, glycine. Those are three letter abbreviated for all amino acids. So remember that the primary structure is like the order of letter in a very long words. If left by chance, there would be 20 to the base or to the power of any number you want for the polypeptide chain, to making a polypeptide chain with amino acids, with those only 20 amino acids. Regions stabilized by hydrogen bonds between atoms of the polypeptide backbone. So the secondary bonds found in most protein consists of uncoiled folds in the polypeptide chains. Most proteins have segments of their polypeptide chains repeatedly coiled and unfolded in patterns that contribute to the protein's overall shape. These coil and folds collectively refers to as secondary structure. Results of hydrogen bonds between the repeatedly constituents of the polypeptide backbone, not the amino acid side chains. Within the backbone, the oxygen atoms have a parallel negative charge, and the hydrogen atom attached to the nitrogen have a partial positive charge. So the coils and folds of secondary structure result from hydrogen bonds between repeating constituents of the polypeptide backbones. Individually, these hydrogen bonds are weak, but because they are repeated many times over a relatively long region of the polypeptide chain, they can support a particular shape for that part of the protein. One such secondary structure is the alpha helix, a delicate coil held together by hydrogen binding between every fourth amino acids. The other secondary structure is beta pleated sheet, two or more segments of the polypeptide chains lying side by side called beta strands are connected by hydrogen bonds between parts of the two parallel segments, and this form of secondary structure of protein we call beta pleated sheet, and the other is alpha helix. For the tertiary level, three-dimensional shape stabilized by interactions side chains. While secondary structure involves interactions between backbone constituents, tertiary structure is the overall shape of polypeptide resulting from interaction between the side chain R group of the various amino acids. The interactions between the side chains involve several types of bonds. We have covalent bond, hydrophobic, van der Waals, hydrogen bonds, ionic bonds, depend on the R chain or the side chain of the amino acids. One type of interaction that constitutes the tertiary structure is called hydrophobic interaction. As a polypeptide folds into its functional shape, amino acids with hydrophobic nonpolar side chain usually end up in cluster at the core of protein out of contact of water. Hydrophobic. Remember thus, hydrophobic interaction is actually caused by the exclusion of nonpolar substances by water molecules. Once nonpolar amino acid chains are close together, van der Waals interaction help hold them together. Meanwhile, hydrogen bond is between polar side chain. Ionic bond between positively and negatively charged side chain also help stabilize tertiary structure. These are all weak interaction. So for the tertiary structure, we have several types of bond, chemistry bond. Quaternary structure, association of two or more polypeptides, some protein only in some proteins. Some protein consists of two or more polypeptide chains aggregated into one functional molecule. Quaternary structure is the overall protein structure that results from the aggregation of these polypeptides subunits. For example, collagen. This is an example of quaternary structure. Collagen, which is a fibrous protein that has three identical helical polypeptides intertwined into a larger triple helix, given the long fiber's great strength. This suits collagen fibers to their function as it preserves of connective tissue in the skin, bone, tendons, ligaments, and other body parts. Collagen accounts for 40% of the protein on human body. Hemoglobin, the oxygen-binding proteins of red blood cells. An example for globular protein consists of four polypeptides, two of one alpha subunits and two of beta subunits. Both alpha and beta subunits consist of alpha helical secondary structure. Each subunit has a non-polypeptide component called heme. Each subunit has heme with the iron element atom that binds oxygen. Even a slight change in primary structure can affect protein shape and ability to function. For instance, sickle cell disease. This inherited blood disorder is caused by substitution of one amino acid, called valine, for normal one glutamic acid at the position of the sixth amino acid in the primary structure of hemoglobin, the protein that carries oxygen in red blood cells. Normal red blood cells are disc-shaped, but in sickle cell disease, the abnormal hemoglobin molecule tends to aggregate into chain, deforming some of the cells into a sickle shape. So a person with the disease has periodic sickle cell crisis when the annular cells clog tiny blood vessels, impeding blood flows. You've learned that a unique shape endows each protein with a specific function, but what determines protein structure? You already know that a polypeptide chain of a given amino acid sequence can be arranged into three-dimensional shape, determined by interactions responsible for secondary and tertiary structure. However, protein structure also depends on the physical and chemical conditions of the protein's environment. If the pH or salt concentration, temperature, or other aspects of its environment are altered, the weak chemical bonds and interactions within protein may be destroyed, causing the protein to unravel and lose its native shape, a change called denaturation. Because it is misshapen, the denaturated protein is biologically inactive. When it's denaturated, it is now biologically inactive. Protein transferred from an aqueous environment to a nonpolar solvent such as ether, chloroform, those chemicals disrupt the hydrogen bonds, ionic bonds, or disulfide bridge that maintain protein shape. Heat, which agitates a polypeptide chain, for example, in the white of an egg, becomes opaque during cooking because the denatured protein are insoluble and solidify. This also explains why excessively high fevers can be fatal. Proteins in the blood tend to denature at a very high temperature. If the primary structure of polypeptides determines a protein shape, what determines primary structure for proteins? The amino acid sequence of a polypeptide is programmed by a discrete unit of inheritance known as gene. Genes consist of DNA, which belongs to the class of compounds called nucleic acids. Nucleic acids are polymers made of monomers called nucleotides. The two type of DNA of the two type of nucleic acids enable living organisms to reproduce their complex component from one generation to the next. DNA provides directions for its own replication. DNA also directs RNA synthesis. This entire process is called gene expression. DNA is the genetic material that organisms inherit from their parents. Each chromosome contains one long DNA strand molecule, usually carrying several hundred or more genes. So gene expression is this process. DNA transcript to RNA, and then RNA translates to protein. This equation we call it gene expression. When a cell reproduces itself by dividing, its DNA molecule are copied and passed along from one generation to another. The information that program all the cell's activities is encoded in the structure of the DNA. A given gene along a DNA molecule can direct synthesis of type of RNA called messenger RNA, mRNA. The mRNA molecule interacts with the cell's protein synthesis machinery, this is the ribosome, to direct the production of a polypeptide which falls into a part of protein. We can summarize the flow of genetic information as DNA transcript to RNA, then translated to protein. Nucleic acids are macromolecules that exist as polymers, called polynucleotide. As indicated by their name, each polynucleotide consists of monomers, and these monomers are called nucleotides. A nucleotide in general is composed of three parts. Carbon, 5-carbon sugar, we call it pentose sugar. Remember pentose for the 5-carbon sugar. And a nitrogen containing base, nitrogenous base, and 1,2,3-phosphate groups. There are two families of nitrogenous bases. We have a pyrimidine and purine. Pyrimidine consists cytosine, thymine, uracil. Thymine found only in DNA, uracil found in RNA only. Purines are larger than pyrimidine. You see it consists of 2 carbon rings. Those composed of adenine and guanine. The specific pyrimidines and purine differ in the chemical group that attach with it, attached to the ring. Adenine and guanine and cytosine found in both DNA and RNA. Thymine is found only in DNA and uracil is found only in RNA. Remember those. The only difference between these two sugars, remember that each nucleotide composed of sugar, nitrogen, and phosphate group. The difference between the sugars found in DNA and RNA, these two sugars, is that the deoxyribose locks an oxygen atom on the second carbon in the ring, Hence, the name is deoxy. Deoxy, that means minus or lack one oxygen atom. So far, we have built a nucleosides uh, base attached to carbon. These two components, we call them nucleosides, and nucleosides plus phosphate group form nucleotide. When we say nucleoside, we mean nitrogen base with sugar, the pentose 5-carbon sugar. Both of them nucleosides. When they attach with phosphate group, we call this compound or substance nucleotide. We have built nucleoside base plus sugar. To complete the construction of nucleotide, we attach 1 or 3 to 3 phosphate group to the 5-carbon sugar, and we now form the nucleotide. A DNA molecule has 2 polynucleotides or strands. We call them polynucleotide strands that wind around an imaginary axis, forming double helix. We call the DNA molecule that's formed from 2 strands of polynucleotide double helix. The 2-sugar phosphate backbone, so the backbone, this blue ribbon, this is the backbone for the DNA. It's composed for 2-sugar phosphate. So the sugar and phosphate together form the backbone, run in opposite direction from 5 to 3. These 2 strands run in opposite direction from each other. This arrangement is referred to as antiparallel. We call them antiparallel. The sugar phosphate backbone are on the outside of the helix, and the nitrogen bases are paired in the interior of the helix. The 2 strands are held together by hydrogen bonds between the paired bases. We have guanine, cytosine, thymine, adenine, guanine, cytosine. Also, always adenine is bound with thymine, cytosine with guanine, and they bind with each other by hydrogen bonds. Remember that in base pairing, only certain bases in the double helix are compatible with each other. Adenine is one strand, or in one strand always pairs with thymine in the other. Reading the sequence of bases along one strand of the double helix would tell us the sequence of the bases along the other strands, because we know that guanine always bonds with cytosine and thymine with adenine. RNA molecules, by contrast, exist as a single strand. Complementary base pairing can occur, however, between regions of 2 RNA molecules, or even between the stretch of nucleotides in the same RNA molecules. Pairing with an RNA gives it a 3-dimensional shape necessary for its function. Note that RNA it's paired always with uracil, adenine with uracil. Thymine is not present in RNA. So this is the difference between RNA and DNA. Uracil is always found in RNA strands, and thymine always found in DNA strand. DNA always exists as a double helix. This is the other difference between them. And RNA molecules are more variable in shape. Remember these 2 differences between DNA and RNA. And thank you for your listening.